Good morning. Hi. As Francesco already said, I'm a PhD student, and my project it's part of CerebNEST project. With CerebNEST, we are trying to simulate the cerebellar circuit and also his properties by means of large-scale spiking neural network, all implemented in NEST. So. You would ask why to simulate the cerebellum, why it's so interesting to simulate the cerebellum. Well, first of all, from the functional point of view, the cerebellum has a crucial role in motor control, so behaving like a feedback and a feed-forward controller. And it acts in timing and gain of our motor responses, being able to predict and also compensate for possible perturbation. While from an anatomical point of view, it has this, I would say, beautiful modular microcircuit structure that makes it particularly suitable for simulation when one wants to progressively increase the level of complexity or the numerosity of the neural population and investigate the different synapses dynamics, including plasticity mechanism. So here I will give you a quick overlook on the cerebellar structure that we are currently able to simulate. We have 5 main neuronal populations: the granule cells, here, that are the input stage of the cerebellum, receiving input signal from the mossy fiber, the Golgi cell, the red one, that are inhibitory neurons that form this feedback and feed-forward inhibitory loop that regulate the activity of this granular layer. Then we have the Purkinje cell that Jonathan showed you a very nice picture of, that receive excitatory inputs from the parallel fibers coming from the granule cells and inhibitory inputs from the molecular layer interneuron, the orange one. The Purkinje cell also received inputs from the inferior olive through the climbing fibers, and with this type of inputs, the Purkinje cell is able to decode the perturbation occurring and learn to predict it, acting on the cerebellar, deep cerebellar nuclei, that are the output stage of the cerebellum, that are the ones that generates the motor commands response. So it needs to be said that the neurons in the network, in the CerebNEST network, are modeled by fine-tuning the extended generalized leaky integrate and fire model implemented in NEST by matching experimental data, but also former model implemented in Neuron. This makes the network implementation particularly efficient from a computational point of view, and while keeping a certain level of biological plausibility. And a crucial factor also in this microcircuit are the multiple plasticity mechanism happening inside the network to adapt the motor output response. In the real cerebellar circuit, there are many plasticity sites in CerebNEST. 4 of them are actually currently implemented, and there are 4 of them, and there is the one showing in this figure. So once we have this large-scale model of the cerebellum that has also a good computational cost, we can embody this in robots and test its learning capability through different protocols. For example, famous one is the eye blink classical conditioning, where the robot needs to learn to anticipate the motor response coming from the deep cerebellar nuclei to prevent an unconditioned stimulus, usually a nerve puff in the eye. Or, again, a reaching movement perturbed by a force field, where, again, the cerebellum needs to adapt as I was saying before, the gain of our motor response to compensate a contrasting force and still reach the target. Or again, we can challenge it to generate an ocular motor command involved in the vestibular ocular reflex. And those are the basics to understand the CerebNEST project evolution. I give you a brief panoramic view here. So it started with two former PhD students, Alberto Antonietti and Alice Gemignani, that in collaboration with the University of Pavia designed the robotic experimental protocol to test the network, developed the single-point neuron models that would replicate the cerebellar neuropopulations, built microcircuit models and embedded in the robots that are either real or simulated. So you would ask what's left to do. Now there is me and another PhD candidate in our lab, and we are following the project with the aim of improving two main different aspects of CerebNEST. So Benedetta Gambosi, the other PhD student, is working on the robotic embodiment part, trying to test the role of the cerebellum as a forward and inverse model. All of this done in a closed-loop simulation. The system is basically made of a controller, which models the brain, and an effector, which represent the body and the environment. The controller aims at replicating the main aspect of the motor control, so basically planning the generation of the commands and the state estimation. Again, this is a large neural network entirely implemented in NEST, constituted by spiking neurons that are defining either to be biological plausible or to represent a specific function that we need to use to generate the motor control. So, for example, could be these light green blocks that transform a kinematic command to kinematic signal to a dynamical one. The effector models the body, and at this stage, Benedetta is modeling the control of the elbow joint and its position and interaction with the environment. This part is implemented in Bullet, and it connects with the controller through a music interface. So since her goal is to test the role of the cerebellum as an internal model, she is looking at the adaptive capabilities in the prediction of the feedback and the state, and the consequent refinement of the motor commands in output. For this reason, plasticity rules are included in the model, and at this stage, she is focusing on using an ad hoc STDP rule between parallel fiber Purkinje cell and parallel fiber molecular layer interneuron plasticity. The other line, let's say, of improvement is at the microcircuit level. So part of my PhD project is about the development of a diffusive plasticity model that could be integrated in the spiking neural network model for the cerebellum. So we know about the existence of this little molecule, second messenger, that is the nitric oxide for quite a few decades by now. But personal thought, it doesn't have the right amount of attention it deserves, at least in the computation in modeling field. So who is the nitric oxide? It is a very, very small molecule that is produced by the nitric oxide synthase enzyme, which comes in three main forms. One of them being the neuronal one, and has been located in the dendritic spine of certain type of neuropopulation. In the cerebellum has been found to be in the granular layer and in the molecular one, and in particular in the granule cells. So the peculiarity of this molecule is the ability to freely diffuse in the tissue. It's highly soluble in lipidic and also aqueous means. So it exerts a type of communication that bypasses the mere anatomical connection. Let me show you how it works with this very simplistic animation. So let's imagine the stimulus arrive at these 3 points in the network. The nitric oxide synthase will activate and start producing the nitric oxide that diffuse freely, forming this a sort of cloud of influence in which also synapses that were not active will receive the NO signal, let's call it, and they will also be influenced by it. Indeed, there are many experimental studies showing that the nitric oxide is a necessary but not sufficient condition to plasticity to happen. And this is what makes this, for me, very interesting. So combining this, the plasticity rules with this diffusive type of signaling. These are the main steps that we defined to build a model. First thing first, we need the geometry. So we need to know where the structure that will be affected by the nitric oxide cloud, let's say, are located with respect to the nitric oxide sources. So for this step, luckily we have the brain scaffold builder developed by Robin Shepherd in Pavia that allow us to integrate the scaffold reconstruction of a small cerebellar portion with all the neural population that I mentioned before, and also the nitric oxide sources. All, again, simulated with NEST simulator. Then we need a single source model for the production and diffusion of the nitric oxide to be integrated inside the network and eventually including the dependency of the nitric oxide concentration in the plasticity model. So the single source model is pretty simple. We have three equations modeling the production of the nitric oxide and one modeling the diffusion. The first equation expresses the amount of calcium calmodulin complex that is formed after a spike event causes the influx of calcium in the postsynaptic neurons. Then the calcium calmodulin complex activated the nitric oxide synthase, and we describe this activation with the second equation. While the production of the nitric oxide from the nitric oxide synthase is for now just a simple is just a model as to be proportional to the activation of the source. What's left to model is the diffusion that we implement by using the heat diffusion equation from a point source. So thanks to this low molecular weight that I was mentioning and the non-polarity of the nitric oxide, we can safely assume that it diffuses isotropically through the tissue. In this way, we have a constant diffusion coefficient and radial symmetry. So a lot of simplification. Moreover, we need to take into account both the background reaction consuming nitric oxide and its production. So we add two terms to the diffusion equation, the one describing the consumption that is defined by the lambda parameter, and the function describing the production term, that is the system of the differential equation I already described. So we compare the single source model with the simulation run in Neuron using the reaction and diffusion module that implement the same function but with different modeling approach. So in particular, we exploit its ability in simulating with a high level of realism the biochemical reaction taking place in the intracellular space. And here there is an example of the resulting nitric oxide concentration produced from one source stimulated at 100 with 100 millisecond burst at 40 Hz. While here we compare the result for the diffusion from a single source stimulated with a single spike obtained with our implementation, the one shown in green, versus the results obtained by Garthwaite in 2016. So once we have this single source model, we need to define their location in the network. As you can see from the image, we place the NO sources around the glomeruli that are this structure in the granular layer where the connection between granule cell and mossy fiber are formed. And then we simulate the activity of the network, where a bundle of mossy fiber is stimulated and the glomeruli are basically activated by it and will in their turn activate also the NO sources around them. So then we need to integrate this signal, this single source signal, to form the cloud of influence and, or if you prefer, this kind of volume transmitter signal. Then to verify that what we were doing was kind of correct, we reproduce an in vitro experiment where we have a probe able to measure the nitric oxide concentration in a certain point of the network, and we compare our simulation results with the actual experimental data by simulating the same stimulation protocol applied in vitro and including also the specific function of the probe that used to measure the nitric oxide. The very last step, as I was saying, is modeling the role of the nitric oxide in the plasticity mechanisms. There are quite few evidence of the role of the nitric oxide being involved in long-term potentiation or depression of synapses, in particular in the cerebellar cortex, but there are some discrepancies between the different experimental results. What we are planning to do is to test in our network some of these hypotheses to see if the model is able to replicate the relevant feature emerging from this experiment, while also gathering our data in vitro. So we can go on following this double approach, in vitro, in silico experiments. So I hope I give you a sense of what CerebNEST is about, what we are currently working on, and what are the future perspectives of this project. And I thank you, and thank you also for organizing this nice event. Thank you.